Okay, so now we're continuing on with comparing samples. Now in chapter 8, we were comparing two samples to proportions, to means, and to variances or standard deviations. Now in chapter 11, we're going to be comparing samples with three or more samples, and we'll be comparing the means between these samples. All right, so an overview. This technique is known as analysis of variance, or ANOVA, and it's a method for testing the equality of three or more population means. And what the theory behind it uses their sampled variances, thus the name, the analysis of variance. There are two types we're going to look at. First, the one-way ANOVA. In this case, the sample data are separated into three or more groups according to just one characteristic, also known as a factor. And then the second way ANOVA test we're going to look at is known as a two-way ANOVA. In this case, the sample data is again separated into three or more groups, but it involves two factors, and we'll be doing a test for the interaction between the two factors. Then we're going to be testing one of the effects that we'll call the row factor, and then testing for the other effect, which we will call the column factor. First, let's start with the simple case of a one-way ANOVA. The requirements for a one-way ANOVA are the populations need to be approximately normally distributed. They must have the same variance or approximately the same variances. The samples need to be simple random samples of quantitative data. The samples need to be chosen independent of each other. And the samples are from populations that are categorized in only one way. We only have one thing that we're looking at between them. So as an example, again, let's go to your Minitab lab books and look at the example known as, under one-way ANOVA, the weights of the poplar trees. Now we're going to enter our data into four different columns. So enter the values under none into C1. Enter the values that are listed under fertilizer into C2. The values listed under irrigation into C3. And the values under the column heading fertilizer and irrigation into C4. Now we're going to select stat and then ANOVA and then one-way unstacked. Unstacked means we put all of our data into the four different columns. So now we have four different groups that we're going to be comparing to see if the average from them are the same. In this case, the null hypothesis is mu1 equals mu2 equals mu3 equals mu4. The ANOVA test, the alternative is at least one of them is different. The ANOVA test itself does not tell us which one is different. If we reject the null hypothesis, we'll just be finding out that at least one of them was different. You're going to enter in C1 to C4 into the box labeled responses. And then you'll get a test statistic. Remember, with the ANOVA, the test statistic is an F statistic. We have a p-value of 0.007, which is very small. Thus, we're going to reject the null hypothesis. In this case, there is enough evidence to say that at least one of the means is different. You would then need to do some pairwise comparisons to see which one is different. You would then compare sample 1 to sample 2, sample 1 to sample 3, sample 1 to sample 4, sample 2 to sample 3, sample 2 to sample 4, and sample 3 to sample 4. But that's for another time. All right. Next, we're going to go to a two-way ANOVA. In this case, we are looking to see if there's an interaction between two different factors, and to see if the two factors have an effect in the different categories. The requirements are very similar. The data has to come from populations that are approximately normally distributed. The populations have to have the same variances or close to the same variances. The samples need to be chosen as simple, random samples. The samples need to be independent of each other. And in this case, the sample values need to be categorized in two ways. And all the cells have the same number of values. If one has if we have 5 pieces of data for one group, we have to have 5 pieces of data for every group. There are other types of two-way ANOVAs known as unbalanced design, but we're only considering the balanced design right now. So here again, we're going to look at the same example from the Minitab Lab book, but now we're going to categorize our poplar tree weights from 2 different sites. So we still have the same 4 treatments, and now we have 2 different sites. Now this time we're going to enter the data in a little differently. We're going to enter in all 40 data values into column 1. And then in column 2, you're going to enter either a 1 or a 2 next to each of those 40 values, depending on whether they came from site 1 or the value came from site 2. Then in column 3, we're going to enter in our treatments. And remember, we had 4 different treatments. We had none, fertilizer, irrigation, and fertilizer and irrigation. So in column 3, you're going to enter a 1, 2, 3, or 4, depending on whether that value came from treatment 1, treatment 2, treatment 3, or treatment 4. Once you get all of this data entered into these 3 columns, you're going to go to Stat, ANOVA, two-way. You're going to enter in C1 into the response box. You're going to enter column 2 into your row factor, and you're going to enter column 3 into your column factor. You could have switched column 2 and column 3. You just need to keep straight which one you put in as your row factor and which one you put in as your column factor. Remember, for us, the row factor was the site, and our column factor was the treatment. So here are how we read these results. Now, we have 3 different tests that we did. First, we tested for an interaction. Is there an interaction between the different sites and the treatments? Well, in this case, our test statistic F was 0.17 with a p-value of 0.915. That's a very large p-value. Thus, we failed to reject the claim. And our conclusion is there is no interaction between site and treatment. Then we're testing for our row effect. Remember the row it was our 2 different sites. So F, in this case, our test statistic F is 0.81, and our p-value is 0.374. Again, very large. So we failed to reject the claim there is no effect from the sites. Now we test our column effect, which were those 4 treatments. In this case, our test statistic was 7.50, and p is equal to 0.001. In this case, there isn't an effect on the weight from those 4 different treatments. But if you want to see there's a lot of formulas that go with this that Minitab is using. It is not necessary for you to interpret the data from Minitab, but if you want to see how the formulas work, all of that is in the lecture notes. All right, so that's the end of chapter 11.